the physical factors online lab. Specifically, we mean the physical factors that can affect the growth of microbes. So in our case, we're looking at bacteria. Just to remind you from lecture, there are both chemical and physical factors that can affect microbial growth. So the chemical ones that we have on the bottom of this slide, these are all the chemicals that have to be in the agar or the broth that you try and grow bacteria on. And in lecture, we went through the reasons why you need each of these, what the bacteria uses them for. So make sure that you still, when you look at any of these bullet points, that you're able to think of why the bacteria needs that particular chemical. Now today, instead, we're going to focus on the physical requirements, and we're going to look into some lab experiments that can help us analyze this or to characterize each bacteria. So whenever you hear the physical requirements of growth for microbes, there are three that I want you to think of. There's temperature, osmotic pressure, which means how much salt is in that media, and there's pH. Okay, so remember temperature, osmotic pressure, and pH. Now, just a reminder, as we talk about temperature as one of the physical factors, you have to remember that every organism, even you, have a minimum, maximum, and an optimum growth temperature. Now, we call that range of minimum, maximum, and optimum the cardinal temperatures. So if you look at this figure in the corner, that would be a graph of the cardinal temperatures of an organism. Now, why do you think it is that we have that specific range of being able to have our cells survive? Well, the main factor is our enzymes. As we went over in lecture, enzymes are proteins. And as you know, proteins are very sensitive. Okay? The metabolism in a cell is very sensitive. So below a certain temperature, it's not going to have, the cell is not going to have metabolic abilities. The enzymes will basically, picture them shutting down as if you're just freezing that cell. And you have this bacteriostatic condition where you have either slow or no growth. Whereas above maximum, well, now what happens at really high temperatures? Enzymes are proteins. They get denatured, okay? So you end up with having cell death or organism death, and we call that bactericidal, because remember, when you see cytal, that means death, okay? So cold temperatures cause stasis, high temperatures above maximum cause death. Now, with those temperature ranges and cardinal temperatures, you have to realize that different cells are going to have different cardinal temperatures. Okay, different organisms will be slightly different. And you can categorize them based on temperature. And again, a lot of this is review from lecture. The first of the three categories based on temperature are the psychrophiles. And these are the guys that I call the little psychophiles, right? They love really cold temperatures. They thrive at temperatures, for instance, in -5 to 20 degrees Celsius range, so below room temperature, cold. Okay, now whenever we ask where might you find them, we're thinking in terms of your everyday life. Where do you experience cold growth? That would be in the fridge or the freezer. And what they're usually causing in those locations is food spoilage. Okay, now keep this in mind because, you know, so many times people think, oh, my food, my food and drinks are so safe in the fridge and freezer, they could last forever. No, they can't. There are microbes that like cold temperatures that will grow in there. And keep in mind, cold temperatures, we already mentioned, they're just bacteriostatic. They're slowing growth down. They are not killing the bacteria. Okay, so always remember where you might find psychrophiles and what they're causing. The next group of bacteria we have is very significant. These are our mesophiles. And with the mesophiles, remember these guys grow at a range between 20 and 45 degrees Celsius. Now you have to circle, star, highlight that range because that is very significant for two reasons. within that range, the 20 degree Celsius temperature, that's room temperature, okay? 37 degrees Celsius is in there. That's body temperature. So if you think about it, mesophiles are going to thrive at room temperature and body temperature. So these are the bacteria that can easily be human pathogens. Okay, so the bulk of human pathogens will be mesophiles. Then we have the last group or category, which is thermophiles. And it's funny because on a lot of the graphs, they show one of the thermophiles as its own group, but we can still consider the temperature groups as 3 because that last guy is a thermophile. Now, when we talk about thermophiles, think thermo. Thermo means heat. Okay, so these are the bacteria that like high temperatures. They thrive above 45 degrees Celsius. And there are 3 terms that we went over that you have to be very comfortable with: facultative, obligate, and extreme. So when you hear these terms, the easiest one to remember first is obligate. When you are obligated to do something, you're required. So obligate thermophiles, they require high temperatures above 45 degrees Celsius. Whereas facultative thermophiles, they thrive above 45 degrees Celsius. They can survive that, but they can also survive below. So facultative, you'll see growing above and below 45 degrees Celsius, whereas obligate thermophiles will only grow above 45. You'll see them dead below that temperature, no growth. The last category of thermophiles are the extreme thermophiles, and a lot of figures like the one on this page, they show them as hyperthermophiles as well. What that means is they simply like really, really, really high temperatures. So they'll only grow above 65 degrees Celsius. Okay, so the extreme high temperatures, and they won't survive below that. Then we have our friend Serratia marcescens. And what's important about Serratia is whenever you see a picture of red colonies, okay, so we're not talking about the media, the agar turning red. We're talking about the bacterial growth itself being red, like in this figure here. Sometimes it shows more as a pinkish color. That's a significant concept, okay, because that concept is that even though each cell will have an optimal growth temperature, that optimum growth temperature will not necessarily be optimal for each and every enzyme in that cell. So with the example of Serratia's optimal growth temperature is 37 degrees Celsius, which is your body temperature, okay? But at that optimum temperature of 37 degrees Celsius, the enzyme prodigiosin, which gives red pigment to cells, it won't be active. It'll be inactive at that high of a temperature. Prodigiosin is only active when the temperature is below 30 degrees Celsius. So here you see that disconnect between optimal growth temperature and a specific enzyme's optimal temperature. Okay, so whenever you see red colonies in pictures or in our lab, I want you to remember that that'll be Serratia grown at room temperature instead of an incubator, and it's red because at that lower temperature, prodigiosin is active, and prodigiosin is simply red pigment. It's an enzyme that gives the cells red pigment, kind of like earlier in the semester when you saw blue colors for transformation. Now, the second physical factor we went over was osmotic pressure. And whenever you hear osmotic pressure, think of salt amount. Now, osmosis is going to be the movement of water, and it's specifically movement from high water to low water, which is the same as saying low solute to high solute. But keep in mind, we're not talking about the salt or the solute moving, we're just talking about the water moving. Now, when we talk about osmosis, you have to be able to quickly identify what happens in each type of salt environment. So you first have the hypertonic environment. And with hypertonic environments, that means hyper, a lot, a lot of salt. Well, think about when you eat a ton of salt, right? Mouth gets all dried out, or when kids used to, they say kids put salt on slugs and dry them out. Horrible, horrible people. But when you think of hypertonic and salty, think of the cell shriveling up. It's losing water, just like you dehydrate when you eat all that salty food. Well, the cells are losing water in hypertonic environments. Whereas hypotonic, that's the one that I told you to think of, hippotonic. And like a big old hippo, these cells will swell as they're taking in water in that low salt environment. The last one is the ideal, especially in the nursing or medical field, isotonic environments. That's why you'll notice a lot of buffers and solutions in the medical field will be called isotonic, because they are what I call happy equilibrium, okay? There's equal amount of salt inside and outside of the cell, so that cell is not going to gain or lose water. It stays perfectly happy, okay? So isotonic is always the goal. Now, when we talk about salt as a physical factor or osmotic pressure as a physical factor, I want you to circle the number 3%. 3% is your magic number because just about any cell can be perfectly fine under 3%, normal bacteria I'm talking about. And once you go above 3%, that's where you start the category of halophiles. So any bacteria able to grow above 3% salt concentration, they will be labeled a halophile. And just like when we talked about thermophiles, you're going to use the same three terms: obligate, facultative, and extreme. Okay, and as you already know, obligate halophile means that the bacteria will only, it's required to grow above 3% salt. So below that, you'll see no growth. Facultative, on the other hand, these guys will grow above 3% because they're halophiles, but they'll also be able to grow below 3%. So you'll see them growing in all of the percentages, except for when it gets too high for them, such as 20-30% salt. Only extreme halophiles will grow at that level of salt. Okay, so extreme halophiles, those are the ones that you'll see growing above 30% salt. And sometimes you'll see slightly different categories listed online for extreme halophiles. Some places categorize it above 20, some above 30, but just think of it as a much higher salt concentration than the normal 3, 5, even 10% that will show halophiles growing at. The third physical factor affecting growth is pH, and this is an important one, especially with regard to how we stop bacteria or try and kill bacteria. So most bacteria, just like most of your cells, will prefer a neutral pH of 7. You then have the categories of acidophiles and alkalophiles. So acidophiles, when you hear acid, that automatically tells you that it's going to be a low pH. Okay, pH below 7, which is acidic. And when you think of acidic locations, especially in your body, that you might find bacteria, the big one is going to be your GI tract. Okay, so when you think of acidophiles in your everyday life, think of the bacteria surviving in your GI tract. The other category here are alkalophiles, okay, and thankfully these guys are less common. Now, when you hear alkaline, that means a pH above 7. Yeah, basic, okay? So with the basic pH, that kills almost all types of bacteria. Very, very few bacteria are alkalophiles. And that's very important to us because think about what you use to clean, right? See all over the news during pandemics, what are they constantly saying? Lysol, Clorox, all of those wipes. Those are very basic pH cleaners, okay? So it's very good that so few bacteria are alkalophiles because it means that we're able to use things like Lysol, Clorox, bleach, ammonia, any of the very basic solutions to kill 99.9% of bacteria. Okay? So now when you want to test physical factors of growth, there are the 3 different factors that you would test. The first one is temperature. So what you do is you streak the same 3 bacteria. So in this case, Serratia, Bacillus, Sterothermophilus, and E. coli. You streak all 3 of them on 4 different TSA plates. So those are the regular plates that you're usually using throughout the semester, just regular nutrient agar plates. They don't have anything bad in them. They're great for bacterial growth. Then what you do is you take each plate and you put it in a different incubator. So the 4-degree plate you would actually put directly into the fridge because the fridge would be 4 degrees Celsius. The 20-degree Celsius plate you would then put at room temperature to incubate. 37 degrees Celsius goes into an incubator set to body temperature. And 60 or 65 degrees Celsius plate, that would also be put in a separate incubator set to that high temperature. You would then set up osmotic prep by having multiple different agar plates, and each agar plate is made with a different amount of salt in it. So each semester we use slightly different percentages, but usually we'll have a lower percentage such as 0.85% or 1% salt, even 2% salt. Okay, and like we said, any bacteria should be able to grow there unless it's an obligate halophile, but most regular bacteria grow at those low percentages. And then we have plates where we added extra salt into the agar. So those would be, for instance, 5%, 10%, or even 15 or 20% salt. Then what you do is on each of these varying salt plates, you streak both E. coli and staph, and you see which salt concentrations does the E. coli grow on, where does it end up dying off, and then look at the staph. Where is the staph able to grow on, and which plate does it end up dying off? The last one are the pH preps. And for pH, studying that, what you do is you take 3 different bacteria, just like we showed in the temperature experiment. You put all 3 on the same plate, and you use 2 different plates. Your first plate that you set up is a regular TSA plate. Okay, so that's normal plate that has all the regular nutrients for bacteria and nothing bad in it. That is a plate that usually has a pH of around 7, so it's nice and neutral. Then you set up the same bacteria on a tomato agar plate. So there's tomato juice in that plate. And the reason we do that, if you think of tomatoes or tomato juice, what do you know about its pH? Well, it's acidic. That's why some people get heartburn after eating tomato sauce or drinking tomato juice. So the tomato juice plate represents acidic pH, and you look to see which bacteria grow happily on the TSA plate versus which ones grow on the tomato juice agar plate. And if you see any of the bacteria grows on one versus the other, that tells you an important thing about the pH that that bacteria requires or thrives at. So now I'm going to give you a series of actual results, and some of these are courtesy of my former student David, who had taken beautiful pictures of results week to week. So the first set of pictures, these would be your temperature results that I'm going to have you analyze in the post-lab assignment. So on these temperature plates, each plate will tell you what temperature it was incubated at, either 4 degrees Celsius, 20 degrees Celsius, 37 degrees Celsius, or the high temperature of 60 degrees Celsius. And each one you'll be able to see whether or not there's growth for Serratia, Bacillus Sterothermophilus, which is over here on each of the plates, and then E. coli on each of the different temperatures, okay? So when you see a plate like this, what that shows, you can clearly see no growth versus growth, growth, and growth, okay? So make sure that you're comfortable being able to visualize what's growth, what's not growth, and being able to visualize what does that growth look like? Is it robust? Is it minimal growth? Is it beige? Is it red? Okay, so always be very specific about your analyses. Here I have the temperature results again. These are the same pictures from the previous slide, but I zoomed in so that you can better see each of the results. So here is the 4-degree and 20-degree result. And here is the 37 and 60-degree result. You then have osmotic pressure results. So these are salt plates. You'll notice that this first plate on the left, this is 2% salt versus this plate on the right is 5% salt. Okay? Both of these plates are the same plate except this one has more salt in it, and both of them were streaked with the same E. coli on the left side here versus staph on the right side here. Okay, and when you analyze your results in the post lab, again, please be very specific. Specify the salt content you're talking about, whether it's 2% or 5%. Specify the bacteria you're referring to, whether you're referring to E. coli or staph, and specify the growth. Is it robust, smeared growth, or is it speckled individual colonies that represent a lesser growth? Or is there no growth at all? Okay, did you see any change in the growth? We then have the tomato juice agar results, and unfortunately these plates were damaged, so I don't have a picture of these results, but I drew out what the results were actually looking like, okay? So here you have the TSA plate that was streaked with E. coli, staph, and lactobacillus. And here you have the tomato juice agar streaked with the same 3 bacteria. On the TSA plate, I showed which bacteria grew, and when I wrote no in big letters, that meant that that bacteria did not have growth on the TSA plate. Okay, so pay attention to where you see growth, where you don't see growth, and how these 2 plates differ when you are then analyzing in the post-lab assignment. The last slide that we have here is practice identification, and this is very similar to how we do the lab practicals for this course. So I gave you 4 different bacteria. I gave different growth conditions, and in the post-lab assignment, I'll ask you to do things like identify particular bacteria or explain differences amongst the bacteria. So here we have bacteria A, okay, and that is the reddish growth there. Here, all 4 of these plates are bacteria B, and we have the temperature 4, 20, 37, or 60 degrees Celsius, and I wrote whether there was no growth or growth. And then here at the bottom, we have salt plates with bacteria C and D. Okay, C and D on each one, and I specified where there was growth and where there was no growth, just in case you couldn't clearly see in the picture, because I know sometimes with reflections and whatnot, it's a little difficult to see. Okay, so make sure when the final portion of the post-lab assignment refers to practice identification, make sure you're looking at the right plates, whether it's bacteria A, B, C, or D. And then as always, like I always tell you, I have no life, so if you have any questions whatsoever, just contact me in Remind and I'll help you out or further explain any of the slides we went over. Okay? Thank you and have a great day. Here I threw in one extra slide just so that you can write down your growth observations from the previous slide with the practice identification. And that way it'll make it a little easier for you to distinguish A, B, C, and D when you're trying to answer any of the post-lab questions. Again, contact me if you have any questions whatsoever. Thank you.